Hello everybody, I'm Ryan Donnelly. I'm Professor of Pharmaceutical Technology at the School of Pharmacy here at Queen's University in Belfast. The school is a leading international centre for education and research in pharmacy and the pharmaceutical sciences. And my presentation today will give you a snapshot of some of the work my group are doing on microarray or microneedle patches, in that I will have a look at some of the work that we're doing on high dose drug delivery and how that can be used to target global healthcare challenges. I'm going to talk about conventional microneedle strategies first to set the scene. I'll then move on to talk about how, if we can change our mindset to consider delivery of high doses of poorly water soluble drugs, how we can open up a number of possibilities. I will talk about the design and manufacture considerations associated with high dose drug delivery using microneedles, and then look in particular at various dissolving microneedle strategies that can achieve this. And finally, I will finish on some important patient and translational considerations. So microneedles, as the name suggests, are arrays of tiny projections on a solid patch, and they can be in all sorts of shapes and sizes, and can be made from numerous materials, including metal, elemental, silicon, glass, but most commonly polymeric materials. And what these microneedles tend to have in common is that they tend to be shorter than 1 millimeter in height, and they tend to be arranged on small patches around about 1 square centimeter. So because the patches are small, we tend to only really be able to deliver potent substances with conventional microneedles. So intradermal vaccine administration would be the most common use. Alternatively, we could do systemic administration of a very potent drug like insulin, for example. Or we could do local drug delivery. We want to target a disease or infection of the skin. Microneedles don't cause pain and they don't cause bleeding because they don't penetrate deep enough into the skin. What they do is to breach the skin stratum corneum barrier, and that means that we can deliver a range of different types of substances. And microneedles have been considered in the news throughout the COVID-19 crisis, and I think what this has shown is that there is an increasing need for microneedle products, and I don't necessarily think that is all going to be around COVID-19 vaccines or even the use of conventional vaccines through microneedle delivery. I think that there is a big benefit, obviously, in developing countries to have a heat-stable vaccine that could be administered without risk of needle stick injury by somebody who isn't necessarily a skilled healthcare professional. But I think where microneedles are really going to come to the fore in the post-COVID-19 world will be an administration of medicines that ordinarily require injections. So this could be long-acting contraceptives or antipsychotics. It could be HIV drugs or therapeutic antibodies. So these are medicines that normally the person has to come to a healthcare setting to have administered by injection by a skilled healthcare professional. I think healthcare professionals are going to be spending a lot of time in the coming years catching up with a backlog in normal treatment and diagnosis, and also in routine administration of COVID-19 vaccines. So if we can take the administration of conventional injected drugs out of the doctor's office and put them into the hands of patients in their own homes, then there would be a lot of benefits in terms of reducing healthcare costs, but also reducing inconvenience for patients and the likelihood that a patient could bring COVID-19 into a healthcare setting or could catch it while they're there. And so this opens a range of possibilities, but we will have to innovate in terms of our microneedles to make this possible. So if we're considering delivery of high dose drugs and hydrophobic drugs in particular, and remember that about 60% of new chemical entities tend to be very hydrophobic in nature, then there are a few things we need to consider. First of all, microneedles create aqueous pores in the skin. And secondly, for most drugs, we need tens or hundreds of milligrams per day, not the micrograms that we're talking about for a vaccine. So we need to think about what implications this might have for the patch size of our microneedle system and how easy it might be for patients to actually apply these. Because these aren't going to be a system that's only applied once or twice like a vaccine, we need to think about what might happen if we repeatedly apply microneedles to people's skin. I do think, and I'll show in the talk, that all of these considerations can be overcome, and I think this will give a range of different cost reduction implications for healthcare providers and competitive benefits for industry. But really, as a pharmacist, what I want to see are considerable benefits for patients, and I think microneedles can deliver that. So if we consider first then how to make high loading dissolving microneedles. So what we really need to do, because microneedles are quite small and they might only weigh 10 or 12 milligrams in terms of the needles per square centimeter, then we need to get a high drug loading in there, greater than 50% weight per weight in the needles. And we should use a biodegradable or low molecular weight polymer, because remember, we're not only depositing the drug in the person's skin, we're depositing polymer in there as well. So if we're going to use a typical pharma polymer that's biocompatible but not biodegradable, then we need to have a molecular weight that's less than 60 kilodaltons so the kidneys can clear it. The alternative is to use a biodegradable polymer, something like hyaluronic acid or dextran, for example. The polymer should be a good film former and have a low glass transition temperature. So what this means is that the microneedles will be hard and sharp enough to penetrate the skin without bending. We should dry the microneedles slowly to promote physical stability. The last thing we want to do is trap the drug in the amorphous state and then have it slowly recrystallize upon storage, because that will influence the behavior of the ultimate product. So the height of the microneedles and the number of needles that we have on each array and the actual patch size will all influence the deliverable dose. And if we're depositing the drug as hydrophobic particles in the skin, then that will of course influence the duration of the delivery after we remove the patch, because remember, the microneedles might dissolve in less than an hour. For a hydrophobic drug, particularly one in particulate form, we're not going to deliver anything in the base plate. So we have to load the drug into the needles and get most of it as close to the tips as we can, because the needles may not insert 100% of their length. We also need to make sure that when the microneedles are removed, we don't leave a sticky residue on the person's skin for aesthetic reasons, and we don't want to waste drug if we can avoid it. So the first disease that we want to consider in terms of high-dose, long-acting drug delivery with microneedles is HIV/AIDS. And since the beginning of the current epidemic, more than 33 million people have died due to HIV infection. And in 2019, there were about 38 million people worldwide living with HIV/AIDS and 150,000 new infections in children. The vast majority of people living with HIV are in low and middle income countries. And in 2019, 21.7 million people were living with HIV and accessing antiretroviral therapy. So this is 67% of the total number of infected patients who are actually getting antiretroviral therapy. Between 2000 and 2019, then new HIV infections fell by 39% and HIV-related deaths fell by 51%, with 15.3 million lives saved due to antiretroviral therapy. But there is a considerable pill burden, so people are taking tablets every day, and either for treatment or pre-exposure prophylaxis in high-risk individuals. And this can then lead to what's known as pill fatigue, where people will not be so compliant with their medication, and this can lead to resistance. Long-acting injections of purely soluble or hydrophobic drugs is an alternative method. So in this case, the person gets a monthly or every 2 months injection of the HIV drug as a crystalline nanosuspension, and more than one drug can be administered if we're treating as opposed to trying to prevent disease. Now, microneedles could be particularly advantageous over hypodermic injections, particularly in developing countries, because they could be self-applied by the person and they wouldn't need to go to a healthcare setting, and then they also won't need to dispose of a hypodermic needle. So if we want to then formulate long-acting antiretroviral drugs into microneedle patches, then we take the nano-formulated drugs at high concentration and load them into aqueous gels. We then cast them into a microneedle mold and we dry and add a border adhesive and an occlusive backing layer to form the patch. The base plate should readily detach upon microneedle dissolution in the skin, and then the nano-formulated drug will be deposited in the viable skin layers for sustained release and absorption by the rich dermal microcirculation. So this is the first study that we did, and thanks to PATH and USAID for funding the work, and Janssen for providing rilpivirine. So rilpivirine is a long-acting antiretroviral drug normally given by injection. We took the rilpivirine crystalline drug nanosuspension, we loaded it into the microneedles, and we applied that to the skin of rats. As a control, we administered the same nanosuspension as an intramuscular injection, and we followed the plasma levels over a 56-day period. And we found that the microneedles performed extremely well relative to the intramuscular injection, including in terms of drug content in vaginal tissue, because remember, heterosexual transmission is the most common route of HIV transmission. It's very difficult to do allometric scaling for long-acting drugs because they exhibit flip-flop pharmacokinetics, where overall plasma exposure is dictated by the dissolution phase rather than the elimination phase of the drug. And so it's very difficult to work out a patch size for a person based on rat data. We have had a go at this, and we estimate that for 7 days human treatment, in other words, to maintain the drug above therapeutic plasma levels, we might need a patch between 25 and 30 square centimeters, which is extremely possible in terms of manufacture, in terms of a microneedle patch that size, as we will see later on. The next compound we considered was etravirine, and we realized that nanosuspensions can be quite expensive to produce. So we looked at just including the plain powder out of the bottle into the gel to make the microneedles, and then we also then made a nanosuspension. And we applied both microneedles independently to the skin of rats, and then we looked at an intravenous injection because there is no intramuscular injection product for etravirine. And as expected with the intramuscular injection or the intravenous injection, we got a rapid peak of plasma levels of etravirine, and then they declined over a 10-day period or so. What we did see for both the microneedles containing the powder form of the drug and the nanosuspension was sustained plasma levels above the therapeutic plasma level for around about a month. And we think then that this shows that microneedles can be used in long-acting delivery of etravirine as another HIV compound. The next compound we looked at was cabotegravir. So again, used in long-acting prevention and when combined with rilpivirine, treatment of HIV when given by injection. And what we did here was we loaded in either the proprietary ViiV nanosuspension, and thanks to ViiV, PATH and USAID for supporting this work, we took the nanosuspension, we loaded it into dissolving microneedles, and we also took the free acid powder form of the drug and also the sodium salt and powder form, and loaded those into the microneedles, and we compared that to intramuscular and intradermal injections of the proprietary ViiV nanosuspension. After 28 days, all of the microneedles still had therapeutic plasma levels in the rats. What was very notable was after 7 days, the free acid was similar to the nanosuspension, but the sodium salt had plasma levels very similar to the intramuscular injection and the intradermal injection. So what this suggests is that microneedles are quite a viable delivery system for long-acting administration of HIV compounds. Again, predicting patch size can be a challenge, but we think that for around about 7 days human treatment, the patch size could be in and around about 20 square centimeters for cabotegravir. So again, a reasonable patch size. We then moved on and we considered administration of both rilpivirine and cabotegravir, because this combination can be used for long-acting treatment. And what we found was that for the rilpivirine, the microneedles actually performed better over a 70-day period than the injection in terms of plasma levels. For rilpivirine, for cabotegravir, we saw a similar pattern to what we have seen before. And again, over a 28-day period, the microneedles look extremely promising. We then wanted to simulate repeated application, because we think that microneedles would need to be applied every week or possibly every month. And we looked at a loading dose of rilpivirine and cabotegravir in the rats, and we also then did that by intramuscular loading dose, with each of the top-up doses administered by microneedles. So we had either a microneedle loading dose or an intramuscular loading dose, and then we basically followed the plasma levels after we applied the patches every 14 days. And essentially, by applying microneedle patches for both rilpivirine and cabotegravir, we were actually able to top up the drug that was being cleared from the body. So repeat dosing does look promising, and what was notable was that we didn't see any adverse effects on the rats with repeat dosing, either systemically or locally at the actual site. The second disease we want to consider is lymphatic filariasis. And this is a disease where over 120 million people are infected, with about 40 million people disfigured and incapacitated by the disease. So basically what happens is the person gets an infection of microfilariae in the bloodstream, and they end up then with adult worms in their lymph node. And this causes damage to the lymphatic system, which can impair drainage of fluid from the limbs in particular. And this causes this elephant syndrome in patients where their limbs swell. Whenever drugs to treat lymphatic filariasis are administered orally, they penetrate poorly into the lymphatic system. However, it is known that if you can deliver particles of less than 100 nanometers into the intradermal space or intramuscular space, you can target the lymphatic system. So loading antifilarial drugs, both anthelmintics and antibiotics, that actually target not only the worm but also the bacteria that symbiotically lives in the belly of the worm, then you could potentially have much better treatment of lymphatic filariasis. So what we did here was we loaded the drugs into solid lipid nanoparticles, and we placed those into microneedles, and we applied them to the skin of rats, and then we did an oral control, and we looked at the levels in the lymphatic system. And what we found was that microneedles led to about 7-fold improvement in lymphatic concentrations of the drugs, apart from albendazole sulfone, where this drug is actually a metabolite of albendazole that is only formed during first pass through the liver. So we have been able, in this case, to target the drugs to the lymphatic system and have a profound improvement in lymphatic levels of the key drug substances here. So this means that we have a potential means of targeting lymphatic diseases using microneedles as well. I've been mentioning how, particularly with the HIV drugs, in order to deliver the requisite dose, the patch size is going to have to be relatively large. So what we did was we got 10 human volunteers, and we got them to self-apply either a small 1 square centimeter microneedle patch, or a patch made up of 16 individual 1 square centimeter patches to their skin. And we used optical coherence tomography, which is the optical analog of ultrasound, to study the depth of insertion and the width of the pore created. And we then compared in these individuals the depth of insertion of the multiple patch needles or the width of pore with those created by the single 1 square centimeter patch. And for the large patch, we looked at various different areas on the sides, in the middle, and so on. So the mean insertion depth was not any different for the larger patch, and neither was the pore width created. So what this suggests is if you give people appropriate instruction, they can apply large patches by hand just as effectively as they can a small patch. So large patch application is viable, and we have in the past made single piece microneedle patches up to 25 square centimeters. Most of these medicines will have to be repeatedly dosed using microneedles. So we took a series of nude but fully immunocompetent mice, and we applied microneedles to their skin on a weekly basis for 5 weeks, and we looked at skin appearance. And at the end of the 5-week period, we found that that had not changed. We couldn't see any evidence of polymer deposition in the skin of the mice. And when we measured transepidermal water loss to assess skin barrier function, we found that that was also unchanged relative to control. We then looked at immune response, and in this case we had 2 microneedle designs. So there either was 11 by 11 or 19 by 19 arrays of 600 micrometer needles. And we looked at 3 formulations. So the acid polymer Gantrez S97, the neutral polymer PVP, and then swellable microneedles that didn't dissolve in the skin and would be neutral in nature. We found that there was no immune response relative to control, and needle density had no effect, and there was no significant difference whether the microneedles dissolved or swelled in the skin. And this was measured by immunoglobulin G levels. We then measured interleukin-1 alpha and again found no significant difference relative to control. Needle density had no effect, and even the low pH of the Gantrez system had no significant impact. We also looked at infection as indicated by C-reactive protein, body temperature and weight change, and again there was no indication that repeat application of microneedles caused infection. So I think that microneedle systems are very promising for high dose delivery, whether we're targeting specific compartments in the body or whether we're doing long-acting delivery of hydrophobic drugs. We can deliver more than one molecule at the same time, and I think that such systems really do have the potential to address global healthcare challenges, particularly in the post-COVID era. Of course, we still need to do some work on understanding pharmacokinetics and comparing between animals and humans, particularly for long-acting drugs where flip-flop pharmacokinetics are in operation. We've recently been very fortunate to receive a large grant from the Engineering and Physical Sciences Research Council to de-risk our delivery platforms in accordance with regulatory advice. We basically will show that the delivery systems are safe and that polymer can be cleared from the system readily, and then develop a bridging program that we can offer to any industrial partner and that will be applicable to each drug that they may wish to be delivered, where they only need to show then that administration of their drug molecule doesn't cause any unusual effects. The microneedles field has made considerable strides in terms of manufacturing scale-up and GMP production over recent years. So we're now confident that a product can be manufactured for the market. Ultimately, we will be involved in macaque studies, infection models for HIV, and also clinical trials to show that the microneedles will deliver the molecules effectively in humans, and we're working towards both of those at present. Ultimately, we would expect to see microneedles commercialized within the coming years, and that this will lead to considerable patient benefit. So that concludes my presentation. All that remains for me is to thank the many sponsors of our work and also to thank the members of my group for all of their hard work and innovation in bringing this work to fruition. Thank you.